Good morning, everyone, or good afternoon, everyone. This is a great honor for me to be able to introduce today's speaker, Dr. Baldwyn Torto. Baldwyn is the principal scientist and head of chemical ecology at the International Center of Insect Physiology and Ecology in Nairobi, Kenya, as well as extraordinary professor in the Department of Zoology and Entomology, University of Pretoria, South Africa, which is one of Africa's leading universities. Baldwyn's research interests include chemical ecology of disease vectors, crop pests and beneficial insects, and applications of semichemicals and natural products in pest and vector management. Baldwyn has received numerous recognitions and awards, including awards from ACS and ESA. In 2016, he was elected Fellow of the ESA and is also a Fellow of the African Academy of Sciences. Baldwyn is an associate editor for Proceedings of the Royal Society B, Pest Management Science, and Journal of Chemical Ecology. Baldwyn is actively collaborates and publishes with groups at Penn State University, Rothamsted Research, the USDA ARS, and several universities and national research organizations in Africa. So without further ado, I turn it over to Baldwyn.
Thank you, John. Thank you so much for the generous introduction. That was very kind of you. I'm actually very excited to be invited to make this presentation. And I'd like to take this opportunity to thank Steve Duke for initiating this process, and of course, Kyle Sawyer for making this happen. Like I said, I'm really very excited to make this presentation all the way from Nairobi. It's a little late here, but that obviously is not going to affect my presentation. I'm very much awake to make this presentation. So the title of my talk is actually Innovative Approaches to Exploit Host Plant Metabolites in Malaria Control. John talked about my background in chemical ecology, and so we are very much interested in understanding the interactions between mosquitoes and plants. And this is an area that not so much is known about. And so we've been looking at this for a number of years now and getting some very exciting results. And amazingly, we find that it's actually a gap, as far as a knowledge gap that needs to be addressed. And we are doing that with quite a number of students and postdocs. So my center is the International Center of Insect Physiology and Ecology based in Nairobi, like John said. So what I'll do is give you a little background or highlights of the kind of research we do at ICP before I continue with the presentation. So the outline of my presentation, it's about that I'll talk a little bit about ICP, and then I'll focus on malaria facts, why it's so important that we should work on malaria. The facts are very staggering. And then the vector control strategies and impact on malaria. Like John said, we are the International Center of Insect Physiology, which means that we're a vector biology institution. So I will spend quite a bit of time talking about the vector. And then the development of new vector control tools, which is based on natural products. And development of new parasite transmission blocking tools, again, that was focused on mosquito host plant interaction. And then I'll provide a summary. So here are some of the general facts about ICP. With those two beautiful pictures showing some of the facilities or some of our infrastructure. We're a center of excellence in Africa for research and capacity building for insect science and its application. We are an intergovernmental organization, the charter signed by 13 countries worldwide. We have about 500 staff members from 39 nationalities and several contracted workers. So we have quite a number of consultants. I think about 10 to 15% of staff are the PhD level. And so we actually drive the research. And we come from various countries from across the globe. We have annually about 150 to 180 graduate students, mainly from Africa. But we get students from all over the world. Some come to spend between about 6 months to a year with us. But for the African students, they have to go through the full course of the MScs and PhDs with us. But they are actually registered in, we are not a degree awarding institution. So we collaborate with several universities in Africa and other parts of the world where these students actually get their degrees. And then we are an organization with a unique history because we are the only center in Africa that is focused on insect science. And in 2020, we're going to be 50 years old. So briefly, how we work is that agriculture is actually the backbone of the economies of most African countries. But focusing on agriculture alone, we've realized that that is not a solution to poverty within Africa. Poverty has other components like diseases. So we have to focus also on the health of the farmers, that actually support our economies. And then also look at the environment in which they work. So within the One Health concept, we look at agriculture, and some of the plants we look at are staple foods like maize, sorghum, and so on. Horticultural crops, we look at migrant pests. Currently, one of the biggest problems in Africa is the fall armyworm. We do occasionally have outbreaks of locusts, and so we have to look at that. We are looking at other sources of proteins for the future of the sustainability of the continent. So insects for food and feed, about 500 species of insects are eaten on this continent. But it's important. We have science, evidence-based information about the nutritional qualities and the anti-nutritional qualities of these insects. For animal health, we look at tsetse flies, ticks, and biting flies. And environmental health, we do look at the biodiversity and conservation of some key species within this continent. Bee health, commercial insects as an income-generating activity to supplement the income of our farmers. And then we do, with indigenous knowledge, a bit of applied bioprospecting. And over all this, we have to actually look at climate change and how climate change affects all of this. And when we come to the health of our farmers and then their families, priority, we have to put malaria right at the top. And most of it, we look at infectious diseases like leishmania, zoonotic diseases, emerging ones are Rift Valley fever, chikungunya, dengue, yellow fever, sleeping sickness. And cutting across all this is the capacity building. So the 150 to 180 students that we have, including postdocs, they drive the research. And so we get a very dynamic program here cutting across all this. And then chemical ecology can be actually embedded in each of these themes: plant health, human health, and environmental health. So that keeps us really very busy. About 40% of the students who work on our projects go through the chemical ecology department. So what is our vision for malaria control? Our vision is to develop and contribute effective integrated vector management strategies towards achieving the WHO target of reducing malaria deaths by 90% in 2030 and eradication by 2040. Now, this is the vision, this is the target set by the WHO, that 90% reduction in malaria deaths in 2020. But then, based on the presentation, there are actually challenges. Whether this target to be met by 2030, that is a question. And of course, the eradication by 2040, because we're already seeing lots of challenges that needs to be addressed based on research to ensure that we can actually meet these targets. So here are some of the facts about malaria. In 2018, this is based on the WHO report of 2018. If we look at the map, malaria is predominantly a sub-Saharan African problem. The high and moderate rates are predominantly within sub-Saharan Africa, right here. So as an insect science institution, we have to take malaria as a priority because 90% of the bedding disproportionately affects sub-Saharan Africa. And in 2017, 290 million malaria cases were reported worldwide. There were about 400,000 deaths worldwide. But 90% of the deaths occurred in Africa in children below 5 years old. And then the resources available for malaria research and intervention is about 3.1 billion. This was in 2017, which is not enough because the burden is still there. This is the map. So the amount of money allocated for malaria research and intervention still needs to be looked at. And like I said, there are challenges. In the last 3 years, 2015, 2016, and 2017, there's evidence that malaria control has plateaued. And that means that a lot more needs to be done to move malaria from where it is now, the 400,000 deaths, to a lot less than where it is. Otherwise, the WHO target is not going to be met. So what about the disease? What is it? What is the biology of the disease? I'm going to spend a little bit of time to explain it, and then we can actually understand clearly where the gaps are and what kind of research that needs to be done, why we have chosen to actually work on the vector and not necessarily just the pathogen. So malaria is a mosquito-borne parasitic disease. It's a single-cell pathogen which are usually known as plasmodium parasites and transmitted from person to person by an infected Anopheles mosquito. So the mosquito has to carry the parasite and then it has to bite a human being. So when we look at the biology over here, we find that the development of the parasite requires two hosts, the human host and then the mosquito host. And so if you have an infected mosquito bite a human being, then the parasite specific stage of it, which is called the sporozoid stage, gets into the bloodstream, and then they take refuge in the liver. And that's when they multiply. That is called the merozoid stage. They multiply in the liver. That is the best place for it to hide and to develop and multiply. And then eventually, from the liver, they attack the red blood cells. And then you get rupturing of the blood cells. But some of the parasites at this stage can go into what we call the gametocyte stage, and they become female and male gametocytes. Those are the infective stages where a mosquito bites a human again. So that's biting twice. Bites a human again, picks up these infective stages. These are asexual stages. And then you do get fusion in the mosquito. But this entire process, which I'm talking about, takes about 2 weeks in the human being. And within those 2 weeks, this person who is infected with the parasite doesn't feel any symptoms. It's only after 2 weeks that the symptoms begin to emerge. Now, after the mosquito picks up the gametocyte stages, male and female gametocytes, in its gut, which is this stage, you do get fusion. Now, the importance here is that the mosquito, this entire process of development of the pathogen in the mosquito takes about 12 days. So the longevity of the mosquito is so critical. If the mosquito lives shorter than 12 days, the parasite will not, or the pathogen will not be able to complete its cycle. But if it can live up to 12 days, and typically an adult mosquito can live up to a month. So 12 days, then the entire development will be completed in the mosquito. And then if you bite again, now we have the stage where the mosquito now seeks a host, and that must be a human host, to infect the person with the parasite. And then the cycle continues. So what this means is that in developing transmission blocking agents or tools, the first thing is to have novel combination therapies because of the different stages of the parasite in the human being. Novel combination therapies that can take care of the various stages. And more importantly, the infective stage that the mosquito. Most of the drugs which are out there are not very effective against the infective stage, that is the gametocyte stage, that the mosquito picks up and then continues with the development. Again, there has to be the vector. We can't only look at humans by developing drugs. We also have to look at methods by which we can block transmission of the parasite in mosquitoes. And then we also have to have more sensitive tools for accurate monitoring of vector populations. Vector population is really very important, and I'll show from my presentation what has happened in the past. This is why we should be looking at that. The pharmaceutical companies focus mainly on drug development within the human, but the two are very important. So malaria control is actually pathogen control and vector control. Pathogen control in humans as well as the mosquito, and then the mosquito itself. So what I'll do is to just highlight some of the work that we've done in this area. And in Africa, what about the mosquitoes that are efficient vectors? There are several Anopheles species, but not all of them are very efficient in transmitting the parasite or the pathogen. There are 4 of them: Anopheles gambiae, Anopheles arabiensis, Anopheles funestus, and Anopheles coluzi. And this varies from region to region. But the gambiae is the most important, although in some parts, particularly in East Africa like Kenya, funestus is now becoming a big problem, filling in the gap because of the tools that have been there targeting Anopheles gambiae. And the question is, why are these the most efficient vectors, despite the fact that we have several species of Anopheles in Africa? There are 4 specific reasons why they're the most efficient. They're easily infected with the parasite. They are long-lived. Like I said, Anopheles gambiae can live up to a month. And they usually bite people. And they bite and rest indoors. So these are the characteristics of these 4 species that make them so efficient to transmit the disease or the parasite. Now, the last 2 points, usually bite people and then bite and rest indoors. That knowledge has led to some tools that have been developed in the past to contribute to bringing down malaria infection or the decline in malaria. And these tools are the use of long-lasting insecticidal nets and indoor spraying. Long-lasting insecticidal nets because they prefer to bite humans. So these nets have been developed, impregnated with insecticides, soft insecticides, barytron-based insecticides. And their use has caused Anopheles populations to go down. And then, of course, indoor spraying, because they like to rest indoors. So these vector control strategies have been very effective in the past. And here is it. The evidence is very clear if you look at this chart, that between 2003 and 2015, the decline has been about 50%, ranging from about 800,000 all the way to about 400,000. Just using vector control tools. But there are other interventions which I'll talk about. So there is evidence that vector control using these 2 tools I talked about, which is insecticide-treated nets and then indoor residual spraying, that has contributed about 60% of malaria decline. Although we are about 50% from 2003 to 2015, that has plateaued. We are still losing 400,000. So that plateau, 2015, 2016, 2017, indicates that there are challenges. And I'll actually discuss those challenges as well. Despite all the progress made, every 2 minutes, a child still dies in Africa. That tells us we still have a lot of work to do, and that is not good, to lose a child every 2 minutes. And the WHO predicted in 2015, 4 years ago, that to be able to go beyond this 400,000, 3 times current funding will be needed to reduce mortality by 90% in 2030. This was in 2015, and we are now in 2019, and it's already plateaued. So it tells us that we still need a lot of work to do, and the current funding situation needs to improve. So here it is, if you look at the evidence, just looking at international funding, between 2003 up to 2016, the funding actually went up. You see a gradual rise, and it picked up by 2015 and then plateaued. And again, if we look at the graph at the bottom, insecticide-treated net coverage in sub-Saharan Africa increased. The pattern is very similar to the international funding for malaria control. And then, if you look at population sleeping under treated nets, that also increased. So obviously, these interventions and the use of these tools contributed significantly to the decline in malaria. And then the other interventions, which combined the use of larvicides, then prompt diagnosis using rapid diagnostic tests, treatment with artemisinin-based combination therapies. All these actually contributed to bringing down the malaria situation to about 50%. But why has it plateaued? And that is what we can look at. So we can look at it from the vector point of view and also the parasite. Why the plateau at the vector point of view is that we have insecticide and behavioral resistance to indoor control. Mosquitoes are very smart. They've changed their behavior. There's also insecticide resistance. And if they've changed their behavior, what is happening? They are biting early. There's also a population that only is outdoor and avoid going indoors. So they bite outdoors. And in most parts of Africa, it's quite warm. So most people sit outdoors and go to bed quite late, maybe around 10 p.m. or so. So there's the outdoor biting population and resting vectors that rest outdoors rather going indoors. Then there's the population that actually prefers to feed on animals or can feed on both humans and animals. So they can get the blood from both sources. Then we have secondary vectors also emerging. That means we have to look at this. We have to have tools to target secondary vectors. And then also the challenge in accurately monitoring vector populations before introducing an intervention. If we look at the early biting population, we have the outdoor biting population, secondary vectors. It means we need more sensitive tools that can target these populations. And then why the plateau at the parasite stage? If we look at the life cycle of the parasite over here, most of the drugs that are out there, by the time it gets to the gametocytes, are not really very effective. The ACTs, the artemisinin combination therapies cannot prevent transmission of the infective stages. Like I said, the gametocytes. And of course, there are emerging reports of resistance to ACTs in some places like the Greater Mekong area, Cambodia, Vietnam, and so on. And sooner or later, it's going to get into Africa. Currently, ACTs seem to be working very well, and then the early diagnosis and so on. But very soon, we'll run out of options. And that is the very reason why we have to be addressing these challenges of the plateau. And then there's the persistent transmission from asymptomatics, those who cannot be treated with drugs because they don't actually have any symptoms, but they're actually carriers of the parasite. They contribute to the transmission of the problem. So why I have this here is to show that we need new strategies to block transmission. And I think the focus should be not just on human, where the parasite, but actually in vector. And this is the reason why I'm saying this, is that in humans, if we look at the drugs, there are 3 classes of drugs that have been developed so far. There's sesquiterpene lactones, artemisinin, artemether, then there's sulfonamides, and then quinolines. These are the main drugs that are out there. But in mosquitoes, to block transmission, we just have primaquine, which is also a quinoline, and it works as an endectocide. It is actually not applied directly to the mosquito, but if it's taken by the human and mosquito takes it up, it can actually block transmission in the mosquito. But the problem is that it has a lot of side effects. So it's not so utilized in this part of the world because of the side effects associated with primaquine. So in reality, there isn't that much in terms of blocking transmission in mosquitoes. So that's a gap that one needs to look at, and that is one of the areas that I'll focus on in my presentation. And if we look at all the various groups in malaria, what we've proposed for the next generation called malaria control tools, transgenic mosquitoes, gene drive, there are groups working on that, which has all kinds of connotations efficacy in the field or in the wild and things like that. They are vaccine development. And then there's novel control tools and transmission blocking drugs. Fortunately, my center, we focus on vector biology. And so I'll be able to present a really good story on what we've done on novel vector control tools. And where transmission blocking is concerned, we do collaborate a lot in this area because that's not actually our strength. And if we look at the drugs that are actually out there and they've been effective over the years, they're predominantly plant-based. The quinines, artemisinin, look at the plants they've come from and the derivatives from the quinines and so on. So we cannot discount plants. Plants are so important. And not only that, but they're also important in mosquito survival, which I'll highlight later on. So can we now go back to plants, but not just based on indigenous knowledge or ethnobotanical information, but also plant feeding in mosquitoes to be able to identify potential alkaloids or sesquiterpene lactones that we can actually use as leads or lead molecules for development. And how can we do this? We can do this, and as I'll highlight in my presentation. But first of all, I'll just focus on the development of novel vector control tools based on natural products, some of the work that we've done. And what we did was first to try and understand mosquito ecology again. Can a better understanding of mosquito ecology contribute to developing these novel control tools? This is really very important because when a mosquito emerges, the adult emerges, both male and female have to sugar feed. They have to look for nectar to feed, and that comes from plants. And in the process of feeding, what we've realized is that they don't just take nectar, they actually take plant tissue. Why they do that, we don't understand. But they do take plant tissue, and that sugar, that nectar that they feed on, that sugar source is required for mating, is required for host seeking. After mating, the female now needs blood, and that blood is just meant for egg development. That is the source of protein. So after mating and looking for the host, once the female has actually taken the blood, male, and gets heavy and can't fly too much, it just stays indoors. But before finding a site to lay the eggs, it will sugar feed in the process because it needs the energy to be able to find the aquatic habitat to lay the eggs. The protein is not enough. So plant feeding is so important. Right after emergence, plant feeding takes place. Even host seeking after mating, plant feeding can take place. And then finding an oviposition site, plant feeding still takes place. So why shouldn't we look at mosquito-plant interaction and find out what other components do they take in the process? What kind of secondary metabolites do they take in the process or ingest in the process? So we looked at the ecology again and found that this is really very important to do so. So here it is to summarize mosquito plant. Sugar ingestion plays critical role in longevity. If it stays for a month, it has to have sugar, fecundity, flight capacity, and host seeking. And how do they find these plants? It's based on fluorescence. They use that to locate their sugar sources. If we can tap into that, we can actually find plant compounds that are really very effective in monitoring mosquito populations. And on top of that, we would not need carbon dioxide because they don't need that much carbon dioxide to find the sugar source. But for females, they need that carbon dioxide to find the blood source where they get their protein. But both sexes don't need that much carbon dioxide to find plants. So plant odors are really very important. If we can identify the attractive volatiles, we will be able to come from the preferred plants, not just any plant, preferred plants, we will be able to get a better accurate monitoring tool. We've done a lot of work in this area, including uninfected and infected mosquitoes, and published in various journals, current biology. We've done it in PLOS, we've done in PLOS NTD. We looked at so many other mosquito vectors of disease. But this is just to show you that we actually do have a lot of background working in this area. So how do we do this? We look at the plants mosquitoes feed on in the wild by setting up traps. Trap up the mosquitoes. We use CDC light traps, bait them with carbon dioxide, and if it's some other malaria or disease vector like yellow fever or dengue, Aedes, we use BG sentinel traps. But CDC light traps are really very good for anopheles mosquitoes baited with carbon dioxide. And then we actually do tests for plant feeding using cold anthrone test. That would tell us that they've actually fed on plants. And then we select the ones that are fructose positive. If they're fructose positive, that means that shows recent plant feeding. And then we extract the DNA, and then we amplify, targeting these two plant genes, TRN and then marquee genes. And then we do sequencing. Once we identify the plants that have been ingested in the gut of the mosquito, we now go over and look for the plants in the field, get them identified by taxonomists, and then we do again DNA extraction and go through the entire process of amplification to confirm indeed that this is the plant that the mosquito fed on. So the next question we ask is how did the mosquito find this plant? If it's a preferred feeding plant for tissue, for nectar, how did the mosquito find that? Here's an example. Here are some examples of host plants we found. And then based on this technique that we have for BAC, this is Senna alata, and we do this work in malaria endemic areas of Kenya, not just anywhere. We go to the malaria endemic areas. Here is a case where we have Senna alata, which is a Fabaceae, Asteraceae, Parthenium hysterophorus, which is an aggressive invasive species. You find that some of the plants are invasive species. There's another one from the Lamiaceae, Leonotis nepitifolia, and then Ricinus cuminis. This is just to highlight a few. So once we've identified these, now we have to collect the odors from these plants, and then to find out whether mosquitoes actually do detect. Does the Anopheles female mosquito detect this? Because targeting females actually makes a big difference. Males don't live that long. So targeting females is really very important. So we use female mosquitoes and the antenna of the female mosquitoes. So this is just to highlight this. If we take the case of Parthenium hysterophorus, which is an Asteraceae, it is called the starweed. That's the common weed and farming weed. But here's his cousin. This is Tanacetum parthenium, which is also an Asteraceae, and it's known traditionally as the feverfew plant. So the question is that even with traditional knowledge, we do have plants that are related to what mosquitoes feed on. So we were very excited to find that here is an Asteraceae plant, which is actually in the same plant. Sorry, we have a Parthenium species, which is very similar to this one, which is known as feverfew. So that was really very exciting. Here it is. If you look at a typical homestead in a malaria endemic area, it's an invasive weed all over within the homestead. So what we did was collect the volatiles from this, analyze, and at the top part, what you find is the FID trace, and then the bottom part is the electroantennogram or the biological detector. So we have the chemical detector, which is right there, the flame ionization detector, then the biological detector, which is the antenna of the mosquito. This is platinum volatiles, and here are the compounds that we identified. They're predominantly monoterpenes. Predominantly monoterpenes, a little bit of sesquiterpene there and an aldehyde there, but predominantly monoterpenes. So what about the other plants? We did the same with Leonotis, again predominantly monoterpenes. Senna alata, predominantly monoterpenes. So what we did was now take the Parthenium one to blend, and we came up with a blend, what we call blend C, of the key components detected by the antenna. And then combined that, we did field work on this in various sites in Kenya. If you take the blend and you combine it with carbon dioxide, you find that you do trap a lot more mosquitoes compared to carbon dioxide. And then it's is slightly, not as great as using SOx, which is human odor combined with carbon dioxide. But when we took individual components in the blend, first of all, the blend, we actually put it in the ratios in which they appear in the volatiles or detected in the volatiles by the antenna of the mosquitoes. But then, if we took the single compounds, what I've done is just highlight one component, which is linalool oxide, and actually it comes as a mixture of isomers anyway. The commercial, and so this is the commercial. I'm only showing the E form of it, and of course the furan. But then it does have the C membered ring. We have the furan side as well as the pana. It's just escaped my mind. But then, if we take the commercial linalool oxide, you find that it's actually a lot better than using the blend, which tells us that with the blend, we do have some of the components acting antagonistically to each other. So that tells us that we need to do a lot more work to optimize this. We've got one component which is great, but the other component is not that great. And then for Anopheles fenestus, it works really very well. So this just proves what I've been saying, or what I said earlier, that plant compounds actually make a big difference to mosquito interaction and mosquito survival. And here we have a situation where we've got a compound which works very well for Gambi as well as fenestus. In trying to optimize what we've got, we realize that the blend of 6 components, there be some antagonism between some of the components. But eventually, we're doing some optimization in the field. We came up with these 3 components. Linalool oxide combined with E-beta-ocimine and beta-pinene has given us a very good blend, which is a lot better than testing it against linalool oxide, which is right here, compared to some of the blends that we tested. We also combined this blend with various components that we've found from human odors, like some aldehydes, but that combination was not so great. It's actually mainly the plant compounds all by themselves that work really very well. And here, what we were trying to do was to avoid use of carbon dioxide, because in remote parts of Africa, there's no way you're going to get dry ice. I mean, dry ice usually comes from the cities, and malaria is usually in rural communities. Although we can find small levels of it in cities, rural communities mean a lot. So if whatever we're doing, it matters to look at reducing the logistics involved. And so we, in this particular study, try to avoid carbon dioxide to come up with something that can be used, in remote parts of of various countries in Africa. So we actually published this very recently, sometime last year in Acta Tropica, showing how they work together as plant-based compounds, or plant-based compounds combined with Malaria-based odors. We've also been working, some of our colleagues have also been looking at targeting the gravid females, the ones that are looking for an egg-laying site. And we actually did come out with a plant compound, again, Cedrol, which was from an infusion of a plant, identified and tested in the field to show that it actually works really very well for another species, one of the efficient vectors, which is Anopheles arabiensis. But again, we see clearly here that Anopheles fenestus was not so great. So there is actually that variation in terms of mosquito responses. And that is why we need to optimize combining different plant compounds to find the best blend that can target not just the efficient vectors, but some of the secondary vectors that are out there. Now, there are some groups that are working on formulations, and some of the formulations are already out. They've been tested in the field. These are known as attractive toxic sugar baits. And what is it that we can do with these baits which are already out there? These baits can be optimized. The efficacies of these baits can be optimized. They are natural compounds with some of the plant compounds that we found. And we think that that is the way to go. and this can target the outdoor biting mosquitoes, which at the moment we don't have very accurate tools to monitor their populations. So what I'm going to do now is just focus on a little bit of what we've done on parasite transmission blocking tools with specific focus on mosquitoes. After identifying the preferred plants fed upon by mosquitoes, we decided to look at the survivorship of mosquitoes on the plants in the laboratory to give us a lot more information. And looking at the data, we selected 3 plants: Parthenium, which is the invasive species. Over here is this Rhizomus cuminous, and here is Bidens spilosa. These are another plant that is fed on by mosquitoes. And we found that also based on our DNA work. Now, the survivorship varies. You can clearly see that it doesn't do very well on water. Clearly, one of the plants that stood out very well is Parthenium, the green highlight over here. Rhizomus cuminous has a lot of extrafloral nectar. So it does extremely well, but it's comparable to Parthenium. So the question we asked was that in the laboratory, apart from sugar feeding, what else can we find in the gut of the mosquito in terms of natural products, in terms of secondary metabolites? Because that can tell us a lot of information. How these secondary metabolites interact with the parasite. If you have an infected mosquito or even uninfected, how does it interact with the mosquito in various ways in terms of survival and so on? So again, we looked at some of the key metabolites from some of these plants. Parthenium at the top, this is the mass spec of it. And then we have phenylheptatriene from Bidens spilosa, and then ricinin. Ricinin is really very toxic. But if you look at this graph over here, just feeding mosquitoes on these secondary metabolites, Anopheles gambiae is able to tolerate phenylheptatriene and Parthenium, but it doesn't tolerate the alkaloid ricinin. Now, what is interesting here is that Parthenium is an allergic respiratory problem. It creates allergic respiratory problems in humans. It creates contact dermatitis in humans and livestock. In allelopathy, it reduces agricultural and pastoral productivity. It's responsible for biodiversity loss. It's a big problem in this part of the world. But the mosquitoes can tolerate Parthenium, and yet humans and livestock can't tolerate Parthenium. So that's really very interesting. We just found that this is an uninfected mosquito. And if it can tolerate this sesquiterpene lactone, what other information can we get out of it? Can we develop it into a transmission blocking? What if the mosquito is infected with the parasite? So what we did was to do this kind of study in collaboration with a group led by Roy Diglands, who is currently at the University of Florida. But he did this when he was at Johns Hopkins University. We isolated Parthenium because we detected it in the gut of Anopheles gambiae, and it's a key component of Parthenium hysterophorus. So here is the life cycle of the parasite in the mosquito. It goes through various stages. And so these studies were done in vivo. And what we found was that the Parthenium actually causes exflagellation of the microgamete. Exflagellation of the microgamete. And here is the data. We look at the control. This is it. This is the microgamete. And when mosquitoes feed on a blood that is loaded with small amounts of Parthenium, this is what happens. By blocking this, it means that the parasite is not able to complete its cycle in the mosquito. So that's a clear transmission blocking agent. And then we compared it with parthenolide, which was isolated from feverfew, the same plant, this plant that is used in traditional sources for curing fevers and for malaria. Parthenolide is very similar to Parthenium, which was isolated there. It does the same job. It also serves as an exflagellation tool against the microgamete. And this comparison was also done with artemisinin. This is what is used against malaria parasites. But artemisinin gave the similar results. The differences were not significant. So I'm not showing that of artemisinin because you get similar results as 2 and 3. So we're very excited about this. That just based on mosquito feeding, we are able to identify a compound that it tolerates. But once it's infected, it actually blocks transmission. So can we develop a drug based on parthenin, which is a sesquiterpene lactose? And I had already shown that the previous drugs like artemisinin are sesquiterpene lactose. So there's a lot of similarity there. That not abandoning plants, but looking at it from the mosquito point of view and mosquito interaction with plants. So we published this about a year ago, 2 years ago, the group led by Royal. So like I said, we do this in collaboration with various groups led by Royal's group at the University of Florida. But parthenin has some issues, and this is it. If you look at the structure of parthenin, the antiplasmodial activity has been shown at the gamma-methylene lactone site, which is here. This is where the antiplasmodial activity is here. But then the other side, where we have the alpha-beta unsaturated ketone, that's cytotoxic. So it means that if we want to develop a drug from this, we need to improve the safety profile of this molecule by targeting this side. And this is quite interesting because we clearly see that, or the information that is being available is that the toxicity due to this part of the molecule is because it reacts with nucleophiles, like sulfurhydryl groups of amino acids, inhibiting several enzymes and transcription factors involved in biological processes. That makes a lot of sense because looking at this part of the molecule, this is clearly a part that you can actually have Michael addition. And it's also good because it can be modified based on various reactions. Like I said, it's an Michael acceptor. This can easily be modified. And that is what we are trying to do in the lab using various graduate students to modify the structure of parthenin and other derivatives or other isolates that are similar to parthenin from similar plants to improve the bioefficacy of parthenin. Now, if we look at the antiplasmodial activity of artemisinin, it's associated with the peroxide group. And if we can do similar things to parthenin, I think that would improve its safety profile. But at the same time, we can also modify here and find out, once we modify this part of the molecule, we also need to modify this as, to find out whether, as a control, whether that also influences bioactivity. So the question is, can we improve the safety profile of parthenin through structural modification? And then we can move on to try and understand the target protein for parthenin and derivatives in terms of the malaria parasite. That would now mean that we're actually involved in drug discovery, but then the focus will be not just on mosquitoes, but also looking at the parasite in humans. So to summarize, I think what we've done is that we do have an understanding of mosquito ecology, that it facilitates identification of mosquito-preferred host plant, the exploitation of these natural synthetic volatile chemicals for use in improved vector surveillance and control. We also have additionally found that it facilitates identification and exploitation of mosquito-ingested host plant secondary metabolites, which can be used as potential leads for synthesis of pathogen transmission blocking agents. At the moment, we know a lot about parthenin, but then we have other plants, and we've also identified certain secondary metabolites, which they can tolerate. But the question is, when they are infected, what does it mean? We can also get similar information from males because they are predominantly plant feeders. And like I said, we have found important components in Parthenium historophoros for vector control. And of course, not just the vector, but also for transmission blocking. I'd like to take this opportunity to thank some of the folks who have contributed from ECP, Vincent Yasembe, who's currently a postdoc at the University of Florida in Royal Dinglanson's lab, David Chwasi, Teresa Koi, and a longtime collaborator from Ohio State University, Woodbury Foster, and Royal Dinglanson at the University of Florida. These are our domain statistics. We notice that we have donors all over the globe, including NIH. Thank you very much.
Okay, Baldwin, thank you very much. It's a great talk. I'm going to go ahead and open up the questions. So if you look over in your GoToWebinar panel down towards the bottom, there's a questions box. Go ahead and start typing your questions in there. I believe your name will pop up. If you like, I can include that. I can also include institution to pass that along to Baldwin. So while you guys are typing up your questions, I'm going to go ahead and start off, Baldwin, with a couple questions of my own. So I find it very interesting. It sounds like it's taking a great direction in terms of control of this very serious problem. I'm very interested in the pollination and nectar aspect of it. Do you think there's going to be some avenues there we can really explore in terms of if the mosquito is visiting a pollination or nectar source before it takes off, can we possibly get microbes or other secondary metabolites in there to possibly control the mosquito or the pathogen?
Yeah, I think that that's a fantastic, opportunity to explore, to find out what kind of microbes. I mean, how do microbes contribute to the composition of the nectar and of course, I mean, even secondary metabolites that they ingest. So I think there's a huge avenue here for exploitation. Perhaps the plants themselves may not be the avenue to detect secondary metabolites that are important, but perhaps maybe microorganisms, and then interaction with the microorganisms and leading to certain specific secondary metabolites might give us some information perhaps that they do matter as far as mosquito biology. They're very important to the mosquito, and so that's definitely a good area to explore. I fully agree with you.
Okay, very good. Okay, I'm going to move on to some questions from Colin and Joel Coates groups. We have looked at many monoterpenoids as repellents for mosquitoes at high concentrations and in absence of CO2. Do you have any thoughts on when a monoterpenoid odor would change from attractant to repellent? And I can repeat that as necessary.
Yeah. Yes. I mean, plants produce these compounds, secondary metabolites or volatiles at specific concentrations, and mosquitoes have to detect these. Obviously, the receptors can only take specific levels. So once we test these compounds, and that's why field evaluation is very important, we test them at different doses, and we find that the optimal doses that they respond to. So beyond that, they'll become actually repellent. So we can actually take advantage of some of these compounds also, not just as attractants, but also find or develop repellents which can be used for body protection or personal protection.
Okay, very good. And Colin, you can go ahead and just email back in if that's or if you have further follow-up. And okay, so thank you, good talk. So I guess sort of a follow-up just while we get more questions on the box here. A while back you discussed the cedrol and you did not show CO2. So are you saying that cedrol was an attractant without CO2, or do you need the CO2 in there as well?
You actually don't need the CO2, but what we found is that if you combine with CO2, you do get an enhanced attraction. CO2 is so important, but it becomes more important when it's seeking a host for blood meal. But the cedrol, with small levels of carbon dioxide, you do actually get enhanced attraction. I can verify that, but I think that where these tests were carried out without CO2.
Okay, very good to know. A lot of this stuff I'm actually looking at hopefully making blends for all the mosquitoes we have out at our yard, so that'll be good. Just out of curiosity, while we're still waiting for other questions to come in, do you think your blend, a while back I believe you had the linalool, osamine, and the pinene mixture. Do you think is that going to carry over in other species, or is this a very species-specific attractant?
It is not species-specific, because we do find that even anopheles, when we set up our traps, we do find that we can actually trap Aedes aegypti. You see, it depends on the site. If you carry out the test in the yellow fever or dengue endemic area, you might get a lot more Aedes aegypti than Anopheles gambiae. And what we are finding is that irrespective of the species, it looks like mosquitoes, virtually all these disease vectors, mosquito vectors, actually go for plant sources, and monoterpenes play a huge role in their behavior. So it depends. So that's why we like to screen these things in the specific sites. Like if it's Anopheles gambiae, we prefer to screen in malaria endemic areas so that it basically biases towards Anopheles gambiae. If we test, but we've been screening these things also in other endemic areas like Rift Valley fever, as well as and when it's Rift Valley fever areas, you do get the floodwater Aedes. When it's in the dengue endemic area, you do get Aedes aegypti. But we need to optimize them a lot more for each of these species, specific species.
Okay, very good. Thank you.
I just wanted to say that yeah, when I was talking about linalool oxide, the isomers, that we do have the theranoside and the pyranoside. I got a little confused there. So when we buy the commercial product, it's predominantly the theranoside, and then about 5% of it is the pyranoside.
Very good. Okay, good. Thank you.
Yeah.
Okay, still other questions coming in? I do have one other question, just because so many avenues to take on this.
Okay.
Going back to just the whole idea about microbes, are there any known microbes that are just sort of, I guess, not an insecticide, but very bad for the mosquito? So instead of a secondary metabolite, you just have microbe fighting the actual mosquito?
I really don't know. I don't even want to guess, because I suppose them potentially there will be microbes. I mean, if you're talking about for mosquito control, I mean, there's the larvicide, which is basically Bacillus thuringiensis. But as to the adult, there may be microbes that I'm not aware of. But I think that's an area that needs to be developed, and that perhaps could also be combined with secondary metabolites if they are compatible.
Okay, very good. Okay, Baldwyn, doesn't look like we have any other questions coming in. So I don't know if we want to wait a couple of seconds or if we want to go ahead and sign off on that one. So we'll stay here for a couple more seconds. If you have any questions, go on, type them on in. And then just one final question, I guess, while we wait for that. In the very beginning of the slide, you said eradication. Was that for malaria itself or the vector, or is that, so are you taking that as a combined thing you have to do, meaning both of them, eradicate both of them?
The eradication, the WHO has set 2040 for eradication. That means that you're talking about the disease.
Gotcha.
The disease itself. We can't eradicate mosquitoes. It's impossible. It's impossible. We can actually bring down the population, but we should have mosquito populations that are not infected or and are perhaps carrying really small loads of the parasites and not going to be a problem. So the eradication is more of the disease, but not the vector.
Gotcha. Okay. Very, very good. Thank you. All right, we have another question from Arunas. As I understand, many companies look for molecules with fast modes of actions like pyrethroids against mosquitoes. Do you think ATSB with a slow mode of action would be sufficient to reduce malaria? Thank you for that question.
Thank you very much for the question. This is a very reason why we need an integrated approach for malaria or malaria vector control. And then it would be good to combine some of these new technologies with sublethal doses of some of the pyrethroids which are out there, or the insecticides which are out there. And that is an idea that we've also been looking at, because, for instance, we do have metarhizium anisoplied, which specifically is very good or is effective against Anopheles gambiae, but it's slow acting. Why can't we combine with sublethal doses with pyrethroids? Set up traps and either have it as a killing agent, or we can have mosquitoes go into these traps and get auto-inoculated with these pathogens. But when they get out there, we have to have a mechanism by which that they can mate, but at the same time they cannot transmit. So it means combining it with sublethal doses to weaken the mosquito or to affect it physiologically before it gets out into the environment again will be an idea that can also be explored. and not just using ATSBs, because ATSBs they're slow acting, and so combining with other integrated approaches would do.
Okay, very good. Thank you. Thank you. Okay, that looks to be it, guys. I'm going to go ahead and just say thank you very much to everyone who's logged in to watch the webinar. Extremely informative. Also very much, Baldwyn, thank you very much for an incredibly good talk, very informative. And I look forward to seeing you again and discussing this more. So thank you very much.
Thank you very much. Thanks so much for the invitation.